Atlantic salmon are a part of Scotland's way of life. They bring employment and revenue to local economies. So maintaining their stocks is an important part of the job for managers in the various district fishery boards. If stocks are threatened, some sort of action's needed. First to stabilise them, then to get them back up to acceptable levels.
There is considerable concern about the decline in rod-caught spring salmon. These fish are highly prized, and any change in their availability has major implications both for the fisheries and for the management and conservation of these populations. One traditional approach to such a decline is to plant or to stock out. However, if the adults are only present in low numbers, the availability of suitable material becomes a real problem. This is the current situation for these early-running spring fish, which utilize the headwaters of upper tributaries for spawning.
Whatever techniques are used, it takes a long time to build up populations. If, at the same time, the adult population's decreasing, there'll be less egg material available to use for restocking. All of which leads to a question whose answer is crucial to those responsible for salmon management. How can the supply of broodstock be increased? Catching broodstocks can be difficult, and if it's not successful, it increases the problems for managers, because, again, it means there are less eggs available for restocking. Fortunately, however, unlike the Pacific varieties, Atlantic salmon can survive spawning. The problem, though, is that they only feed at sea. Some do return to the rivers to spawn for a second time within the year of sea entry. Others, usually the larger females, need to spend a winter at sea, building up energy levels ready for the arduous spawning process. Researchers at the Scottish Office Agriculture and Fisheries Department have been tackling the problem, and one of the areas they have investigated is the possibility of Celt reconditioning. If fish can be encouraged to feed soon after spawning, there will clearly be an increase in body fat and protein. They'll mature more quickly and be ready to spawn earlier than if they'd been left to make their way to the sea. As well as increasing the fish's productivity, this technique also short-circuits the various dangers that are associated with migration. Even the process of holding fish in controlled conditions isn't free of difficulties. Fungal or bacterial infections can develop if fish are damaged or injured. The kelts themselves aren't interested in feeding at low temperatures early in the year. On top of that, their digestive capability is very low before and after spawning. Their actual stomach and gut structures are sound, but the necessary digestive enzymes aren't being produced. This holding tank is in the Scottish Office Agriculture and Fisheries Department hatchery near Perth. The fish in it were caught in the River Tilt, 75 kilometres from the sea. They were stripped in November, and the eggs were fertilized and incubated. Regular treatments kept the fish clean from fungal and bacterial infections, and by mid-January, they'd settled down and were beginning to turn silvery. It was at this stage that the encouragement to feed began. Small herrings, sprats, or prawns on a piece of wire were moved gently in front of the fish's mouths. After a while, they began to make tentative movements towards them. It's important to remember that at this time of year, January, February, the water is still cold, and the fish's digestive physiology is at a very low level. About a month later, most of the fish were feeding, so much so that they began to move positively towards food thrown into the tank. As the temperature rose and the days got longer, their feeding behavior became progressively bolder until fish were prepared to move across the tank to attack and eat items of food. Progress is monitored all the time. The fish are weighed and measured to check the progress. They're vaccinated against bacterial disease, and their gills are inspected for evidence of the parasitic crustaceans, gill maggots. If any of these are found, they're removed to prevent problems developing later. The monitoring continues until temperatures are too high for the fish to be handled safely without causing problems. And all the time, they're feeding heavily. In October, the first checks are made for signs of sexual maturation. In males, changes of colour or shape. In females, the softening of the body wall. The checks are repeated weekly until the fish are ready for stripping. All the females and a good proportion of the males have matured. After fertilization, the eggs seem satisfactory. The process of weighing and measuring continues to establish how much growth they've made since they were captured from the wild. The weight loss at stripping of the females can also be used to determine the weight of the eggs they've produced. When the eggs are eyed, they can be handled gently without fear of high mortality. They are mechanically shocked to kill any unfertilized ones, then counted and sized. When the yolk sac indicates that the fry are ready to start external feeding, samples of each batch are put into tanks for on-growing. After some 4 to 6 weeks feeding, their success in changing from a yolk sac to an external supply of food can be estimated. When they're feeding and growing successfully, the reconditioning process can be evaluated. It's a fascinating and highly practical piece of research aimed directly at helping to resolve some of the issues that confront those who have to make decisions on the management of fisheries. It has shown that Atlantic salmon kelts can be taken from the natural stream and river environment, held in a tank, and encouraged to feed in fresh water. They can mature again without ever having to go to sea. These reconditioned or rematured kelts will then produce fertile ova and sperm to ultimately provide stocks to supplement populations where the numbers are falling. The work isn't yet over. The reliability of the techniques is still being determined, and the quality of the resultant eggs and fry continues to be monitored. When this has been properly established under practical conditions, the researchers will be in a position to evaluate the benefits and costs of Celt reconditioning. All this essential research is supported by the Tay District Salmon Fisheries Board.
This has been a very exciting project for us. It started really as a small pilot experiment. It's really taken off far more than that. It won't obviously produce vast numbers of springfish. It's not designed to do it. It's not on that scale. But it certainly will produce some springfish to the system. But most importantly of all, it is safeguarding the genetic component of the spring stock. And that at a time when really that stock has been under pressure for 40-odd years is very, very important indeed.
It's another example of the direct, practical involvement of the Scottish Office Agriculture and Fisheries Department in fishery management activities. Their investigations and understanding of the processes underlying the fish's life cycle and the environmental factors that affect it equip them very fully to act as advisors and partners in the field of stock and resource management. In a context where even the smallest variations can become critical, the application of informed, well-researched techniques can be the difference between crisis and survival.